Thank you very much, Corey, for this invitation. I would also like to, greet everyone in Munich as well. It's really a great honor to be here. And, I'm excited to actually share the, some very established data, but also some really unpublished and new data from the lab. And I really wanted to start, let me just make sure I have, okay. So I really wanted to start with explaining what really inspires and drives my curiosity to do this research, right? So in general, I'm interested in understanding the molecular or epigenetic basis of psychiatric disorders, in particular anxiety and depression. And I think everyone in this room and everyone in Munich knows really well that both genetic factors and environmental factors, in particular stress, right, play a very important role, right? We know that nowadays we have this biological substrate that is called the epigenome. And I think that people, in both rooms, actually here in Montreal and also in Munich, know that now epigenome can actually provide the substrate through which, let's say, stress and genes can interact to change gene expression, brain structure and function, and contribute to anxiety and depression disorder. There's a lot of animal research that shows that stress can change the epigenome. We have some correlational studies showing that there are epigenetic changes in many psychiatric disorders, but I believe one thing that hasn't been really addressed is that these disorders are very sex-biased, right? They're at least 2 times more frequent in women than in men. And, there has to be some etiology behind this, and there has to be some kind of a mechanism that, drive this. But we know that a lot of research was done in males only, that there is a very, few studies in females, and also that women's health has been understudied, right? So what I would like to invite you today, if we can actually take into account the ovarian hormone status as the third factors that interacts with genes and the environment, and how this can actually, affect the epigenome to, at least in part, explain the, sex bias that we are seeing in anxiety and depression, and hopefully help us, get to some new treatments, right? And let me first explain to you why I believe we should look at the ovarian hormone status. So this was actually the graph that I saw that kind of completely inspired, a lot that is done in my research, right? So people typically tell you that women are at 2 times higher risk for anxiety and depression, but you rarely hear that this is not throughout life, right? This is literally only during the reproductive period. Because if you look before puberty, boys and girls have very similar risk, and you're seeing depression risk in the upper panel, and in the lower panel here, you're seeing how ovarian hormones are changing across the lifespan of women. So here you see that boys and girls pre-puberty have a very similar risk for depression. Then the risk really jumps to 2 times higher in girls when they start menstruating, when you start seeing these ovarian hormone fluctuations, right? And then throughout the reproductive period, when you have these fluctuations in hormones, this is exactly when you see this increased risk in women. The risk further jumps during the perimenopause, right? Actually, this is the highest that you can get during this period when you have these most extreme fluctuations. And then post-menopause, right, the risk actually becomes very, very similar between women and men. I do want to actually emphasize here that we are not really talking about high or low estradiol or progesterone levels. We are talking about these fluctuations in hormones, right? And we know that there are other, conditions in psychiatry, such as postpartum depression, that affects at least 20% of people, when you have this huge drop in ovarian hormones. There is premenstrual dysphoric disorder that affects 5 to 8% of menstruating individuals. And then there is something that also people talk very little about, and this is called the premenstrual exacerbation. So at least in 50% of women, there is this worsening of symptoms premenstrually, even if they are in antidepressant treatment. So I would like to claim that ovarian hormone fluctuation is a major psychiatric risk factor, but it's again, unfortunately, it hasn't been addressed much because it's a female specific. But I do believe that we have here really a lot of opportunity, right, to actually find some treatment that could be sex informed that we haven't actually have at the moment. So now, why would ovarian hormones even be important, right? Because you may think about them as reproductive hormones. But we know that, for instance, estrogen is a very potent neuromodulator, right? So it can affect serotonergic system and noradrenergic system in a very similar way that you see with SSRIs and other anxiolytic and antidepressant drugs, right? Estrogen can increase BDNF mRNA levels. So this is all in line with the antidepressant-like activity. And then people can again ask you, if estrogen is almost antidepressant or anxiolytic, why would women be at a high risk? And again, I want to bring you back to this idea that we are not talking about high or low levels. We are talking about withdrawals or this drop of estrogen or ovarian hormones. They may increase anxiety or depression risk because it requires this periodic or neurochemical adaptation. And my lab would actually like to understand the molecular mechanism behind this, so that, as I said, so we can think about new treatments. So ideally, you would study this in humans, and I want to be inclusive here and tell you that what we study is not only applicable to women, actually it's applicable to all people that menstruate. This can be cisgender women, this could be non-binary individuals, or it can be transgender men who menstruate, right? And we know that, for instance, in humans, you have this follicular phase when there is a high estradiol and low progesterone, and there is a luteal phase when there is a lower estradiol and high progesterone. So I'm going to show you some data from the post-mortem studies in humans, but like primarily in the lab, we actually work with rodent models. And we can use mice because mice have something very similar. It's called the estrous cycle. And it's actually very similar to the menstrual cycle in humans, but it's actually shorter. It's 4 to 5 days. And we typically focus on proestrus when you have high estradiol, low progesterone, similar to follicular phase in menstruating individuals, and early diestrus when you have low estradiol and high progesterone, which is actually more similar to the luteal phase. The way how we do it, we actually take these vaginal smears actually daily for at least 3 consecutive cycles. And then we want to make sure that we know exactly how they are cycling. And I just want to say that, for instance, based on this vaginal cytology, you can tell whether, like, animals are in this proestrus phase or the diestrus phase. And we check their hormones just to make sure that we can really predict based on vaginal smears where they are. And for the majority of the talk, at least the first part, I'm going to show you the data that actually shows these diestrus and proestrus females, right? After 2 weeks of, like, let's say, following their cycle. And then we always actually also, test them with age-matched males. These are all young adult rodents. And then you're also going to see some data where we remove the ovaries so that actually then we can kind of try to rescue the phenotype. And when we started, really, first what we wanted to show in the lab, like, that there are really behavioral changes that are dependent on the cycle. I think everyone knows here, we are talking here about very simple tests. These are anxiolytic, anxiety-related tests, where mice typically spend more time around the corners, in the dark compartments, and in the closed areas. But if you give them anxiolytic drugs, like, for instance, they will be spending more time in the center, in the lit area, and in the open and elevated areas. So what was really, remarkable to us, it's like when we looked at these data that, like, where we have our high estrogenic, low estrogenic females and males, high estrogenic females look more like males. And whenever you have this drop in estrogen, this is when you are seeing this increase in anxiety indices. And I want to actually emphasize 2 things from this graph. This is across all the 3 different tasks. If you put together these 2 female groups and compare them to males, you lose the sex difference. There is nothing, right? So it's a dynamic sex difference that you can actually catch because if you are segregated animals based on the cycle. Another thing I do believe, although, and I'm going to go back to this idea later in the talk, that this is translationally relevant, regardless how simple these tests are, because this drop in estrogen is really also what, or ovarian hormones is also something that we are seeing in humans as something that can increase the risk. So now Devin, my graduate student, when he joined the lab, we wanted to actually understand how these look across the entire cycle. As I told you, typically we look at the proestrus and diestrus, but then there are these additional, phases in between. And it was really interesting to see that our, elevated plasmase test data look almost identical in estradiol curve. And so what you can see also, like, if you take these low estrogenic females, they're these cycling females, right? Not over-optimized, but if they're cycling females, you give them estradiol and test animals after 4 hours, you can actually see decrease in anxiety indices. You can literally mimic the high estrogenic phase by adding estradiol. And then another thing that we did, Layla, my graduate student, actually, she did the overoctomy and then add estradiol. You can't do in 4 hours, you cannot change the behavior. But if you do multiple injections of estradiol, you can again decrease anxiety indices. So the idea from this slide is that, at least in our Black 6 mice, anxiety-like behavior are primarily estrogen-driven. But I don't want to say that progesterone doesn't play a role. I'm just, like, saying it is primarily estrogen-driven because the cycle is always very complex. So now we also had to understand which brain area we wanted to study, right? Because you could study different areas. But we decided to look at the ventral hippocampus because there was a lot of evidence from the mouse model that actually there is this functional distinction across the ventral dorsal axis, right? So if you, for instance, lesion or optogenetically activate the ventral hippocampal neurons, you will change the anxiety-related behavior, but not spatial memory, right? And vice versa, right? So we decided, like, let's work on this, because the idea was like that these neurons actually might be changing with the cycle, and then they might be actually driving those changes in behavior. Another thing that we also saw is that there was like change in the dendritic spine density across the cycle. So during the high estrogenic phase, you have this increase in dendritic spines, and then there is a drop in the dendritic spine, in the low estrogenic phase. Again, why is this important? A lot of people would do studies only in males. And what they would do, they would stress males, see decrease in spines, then they would give ketamine or other antidepressants, see increase in spines, right? And say this neuroplastic effect of antidepressant is actually something probably important for antidepressant-like activity. What you're seeing here in females is that this actually fluctuates naturally, right? And this is where I think it's at least part of this vulnerability that we are seeing in female, that this fluctuation in structure, it's actually also something that at least in part underlies these changes in behavior. But as I said, like I wanted to understand, because I'm a molecular neurobiologist, I really wanted to understand the mechanism of this, right? And so when you're thinking about estradiol can function through two different pathways. One is this classical nuclear receptor bound pathway where estradiol binds to the nuclear receptors. And then when it binds, estrogen receptors actually get to the something that is called estrogen response elements in the genome. And this can change the transcription. On the other hand, estradiol can also bind to the membrane-bound receptors, activate certain kinases, and then there is also a possibility to indirectly change transcription, but typically this is through different transcription factors. So please keep this in mind, because I'm going to come back to this idea a couple of times in this talk as I'm trying to understand the molecular basis of like what we are seeing. And we know that, for instance, in our ventral hippocampal neurons, we did a confocal analysis. We know that both nuclear and membrane-bound estrogen receptors are present. So you could possibly have both pathways being involved. But as I said, like, my expertise actually is a neuroepigenetics. So my question was like, is this actually regulated through epigenetic regulation or chromatin regulation, right? And again, I know that we have a lot of people here that study epigenetics, but I'm going to make sure that everyone is on the same page. When we talk about chromatin regulation, we really talk about this very specialized structure where the DNA is wrapped around the histone proteins, right? And this is important because its opening and closing of chromatin, right, can actually change gene expression, right? And when we started, there was this idea that in post-mitotic neurons, as your hormones are changing, maybe there are changes in opening and closing of chromatin. Of course, I came here. I hope this actually kind of confirmed that we succeeded in proving this hypothesis. But at the time, when I was starting my lab, there was really no data that would actually support this dynamism that would be female-specific and change with hormones, right? Because we are talking about post-mitotic neurons, and a lot of studies that were done previously were done actually in cancer cell lines, right? So, and the idea was, and I'm going to first talk about this first level or 1D genome, 1D chromatin organization. Like, as I said, like the idea here is, let's say, if you are opening or closing the chromatin, you can actually allow or inhibit the binding of transcription factor. This will change gene expression or transcription. And I'm going to first show you the data from the two different assays that we are using. One is called the ATAC-seq, and another called is RNA-seq. So RNA-seq, I think everyone knows because it's a gene expression very well popular. But I do want to just explain, like ATAC-seq, just to make sure that everyone can follow this. So ATAC-seq is this method that actually allows you to see which regions are open or closed throughout the genome. And in this case, we are looking at our diestrus and proestrus. It's the two female groups that I told you about, males. We are looking at the ventral hippocampus. So what we do, we sort nuclei, and then we actually specifically sort neuronal nuclei based using FACS sorting, right? So neuronal nuclei has this new N marker that if you use the fluorescent antibody against this marker, you can actually just purify the neuronal cells. So you increase this specificity or of the assay. And then you use this enzyme that is called transposase that can cut the DNA only if chromatin is open, right? And then if it cuts chromatin, then you kind of amplify it, you do next generation sequencing. When you do bioinformatics analysis, whenever you see this, like this peak, that means that chromatin was open in this area. And when you see no peak, that means the chromatin was closed. And I'm going to just remind you, we are looking at the ventral hippocampal neurons throughout the genome, right? So see like where are the changes, right? So the first question was like, when we do this in our three animal groups, where do we find the changes? It was really remarkable to see that there is almost 30% of genomic region that change chromatin organization, opening and closing across the estrous cycle, right? And that this degree of change that we are seeing is actually very similar when you compare each of these two female groups to males, right? So it's really like something that was very remarkable to us when we saw this data for the first time. The genes that are being affected are very important for neuronal function. You can see the terms such as synaptic transmission, membrane potential, synapse, right? And just to give you the idea, like this is the gene that is actually encoding the AMP receptor subunit. You see more open chromatin. We have more open chromatin when there is like, high estrogenic phase, and then this chromatin actually closes during the low estrogenic phase, right? And so something that I would like you to take from this specific slide is that we have extensive changes in chromatin across the cycle, that these changes are affecting the genes that are very important for neuronal function, and that they're actually also associated with changes in gene expression, as actually shown here on the right. We also saw the changes in the serotonergic synapse-related genes. And so you can see, for instance, here, this is the receptor for serotonin, the gene for serotonin receptor 2 beta, when you have more open chromatin during the low estrogenic phase and the more closed chromatin during the high estrogenic phase, and this is associated with changes in the gene expression. I already told you at the beginning that estrogen can change the serotonergic function, but this is the first time that now we have the molecular mechanism that these chromatin changes are actually something that may actually mediate the changes in serotonergic function and behavior that we are seeing. So then another thing that we did also was like asking the question, how many genes change expression across the cycle, right? And I have to say that we found around 200 different genes that are changing the expression. At least 50% of them had changes in chromatin, which actually showed that chromatin changes are important for driving changes in gene expression. And there were a lot of these genes that were, important for behavioral fear response, something that we expected, hormone-mediated signaling, but there was something that we did not expect, right? Everything that you see here, all of these genes are actually encoding for these ATP-dependent chromatin remodeling factors, right? So there is in order to open and close chromatin, you need these factors that are going to slide the chromosomes and like histones and stuff. And so now we are also seeing that during, let's say, high estrogenic phase, there is also change expression. So not only that chromatin organization changes, but the expression of chromatin regulators changes across the cycle, implying that there is really something about chromatin regulation that is regulating neuronal function across the cycle. We could also ask the question, is this mediated through nuclear or membrane-bound receptor, right? So let's say you take the high estrogenic phase, and then you take your ataxic and RNA-seq data and ask, when I look at what's specifically open or expressed during the high estrogenic phase, am I seeing the enrichment for estrogen response element or enrichment for something else? So basically, we take all of these proestro-specific regions, and then what we actually do is that we actually look at the enrichment for what is the specific transcription factor, actually. And it was really important for us to see that there was no enrichment for estrogen response elements, but there was the enrichment of this EGR1 that actually I just want to focus on, because we see the enrichment of these regions both in the ataxic and RNA-seq data. Why is EGR1 important, right? First of all, we see that it's actually estrogen dependent, right? So we see that actually there is also induction of this gene across the cycle, as well as opening of its regions across the genome. And there was something else that was important about it. It's an immediate early gene. And there were two groups before us that actually were working in male mice, but they showed that actually these immediate early genes, such as CFOS, can be actually candidate chromatin regulator. What this means is, like, when, for instance, your chromatin is closed, you need something that actually would come and specifically open the chromatin, right? And so we actually hypothesized that maybe EGR1 is something downstream of estradiol that can actually do this. And so for now, what the data that I'm showing you actually are consistent with this idea that what we are seeing is actually happening during the through the membrane-bound pathway, and that EGR1 seems to be downstream of estrogen and probably opening the chromatin and changing gene expression. I also want to put you back, because sometimes people are, like a little confused because I show this genomics data and then they're like, but what about, what about behavior, right? So I'm going to just give you an example of a DLK1. This is a gene that is just like a simplified version of my framework so that you understand what I'm talking about. So for instance, this gene changes expression across the estrous cycle. But there were studies before ours in male mice that actually were showing if you knock down this gene, you're seeing increase in anxiety indices in all of these tests that I showed you that change across the cycle. The idea here that in female mice, you don't need to knock down the protein. right? You actually literally can see this fluctuation in the expression that actually can lead to these changes in behavior. And we see that, for instance, during the high estrogenic phase, there is like a more highly expressed gene, and then it actually kind of down-regulates during the low estrogenic phase. This is associated with the more open chromatin during the high estrogenic phase than during the low. And there's an EGR1 binding motif. So the idea here is that EGR1 would be induced during the high estrogenic phase, open the chromatin, change in expression, and change behavior. So that's something that I wanted you to actually think about. However, I was talking to now just about this 1D chromatin, but there is something that is also called 3D genome, right? And this is this higher level of organization that it's almost like a black box in neuroscience because we know very little about it, but we know it's important for gene regulation, right? So for instance, when you look at the chromatin, there are these loops that actually brings things in the genome that are far away together that are also important, right? And this is how you have these enhancer promoter interactions that sometimes it's not really visible in the linear genome. There are also this higher level of organization, such as compartment, right? I think everyone here heard about euchromatin, which is typically associated with the open, and active genes. And then there's like this more close chromatin, close to the nuclear lamina, that's more associated with the inactive genes. So the way how we do it, there is this assay that is called the Hi-C. It's a kind of very complicated, so I'm not going to go into the detail, but you literally cross-link these 3D genome interactions, and then you can actually do the sequencing and find them out, right? And then we combine this data with our ATAC-seq and RNA-seq data to actually find how important this is for gene regulation across the cycle and between the sexes. This is how the data looks typically. Each of these dots is one of these 3D genome interactions. And then you see this is like open region or urochromatin. Then this is a closed or inactive compartment. And then I'm going to show you some data on the compartmental organization, which is typically what's shown here. And then some data on these enhancer promoter interactions that are typically this kind of a long range loops that are being formed. So the first question was like, if you look at across the cycle and between the sexes, do you see any changes in compartment, right? And importantly, we saw only changes in X chromosome. So now this may not be very surprising because I'm just going to remind you that females have typically 2 X chromosomes. One of them is typically inactivated and closed in chromatin conformation, while males have this active, 1 X chromosome. So even if you are really never seen this data, you look at the low estrogenic females and males, you're like, they're different, right? Of course. But there was something that we did not expect, and this is that as you are seeing, like for instance, this region here, as the estrogen levels rise, you are seeing like that some regions are becoming more similar to males after high estrogenic phase than to females, right? This is only in the X chromosome. It's typically always going this direction. You see more estrogen, there is more similar to males. But there was also something very interesting because typically what you see that different chromosomes do not interact with each other. It's only really within the chromosome that you have these 3D genome interactions. But during this high estrogenic phase, we saw that basically the X chromosome might be increasing the interchromosomal interactions. And for us, it almost seemed like maybe there is this inactive X chromosome, which you typically think about, okay, so it doesn't do anything in cell, right? Except that there are some few SKPs. But we actually had this kind of indication that there might be a decondensation event happening during the proestos, which was really one of the most radical findings. And we wanted to actually follow up on that. So what we did, we took some of the regions that we show a lot of changes in compartments in the Hi-C data, and we fluorescently labeled them, right? So then what we did, we took the ventral hippocampal brain slices, and we stained them with these fluorescent antibodies so that we can actually microscopically now measure the distances, right? And we were able to show that, for instance, if the 2 probes were more close during the low estrogenic phase, they physically shift and they become more similar to the males during the high estrogenic phase, right? You may also ask the question, are there changes in the gene expression? Not too many, but we also saw some genes such as this serotonergic receptor-related gene that actually changes the compartmental organization and then becomes more similar in the expression during to males during the high estrogenic phase than during the low estrogenic phase. So what we are actually showing here is that there might be something that I like to call the estrocycle stage-dependent SKP genes, right? So I just want to remind you, people show that there are SKPs that are tissue-specific, SKPs that might be developmental window-specific, but that there are such a dynamic SKP genes that no one has shown so far. So we are now working actually on this. And so I want to show you one more piece of data that actually shows this dynamism that is specific to inactive X chromosome, because people typically will see this data and they know that if you do genomics assay, you can't really distinguish between active and inactive X chromosome. But we could do something that is called the X chromosome painting that I'm going to show you. So in order to inactivate the one of the X chromosome, you need this long non-coding RNA that is called X-cyst, right? And typically, inactive X chromosome is coated with this X-cyst. So what we decided to do, we decided to actually do the X chromosome painting together with this X-cyst RNA fish. And this actually allows us to mark the inactive X chromosome and then to see whether there are really changes in volume during the estrous cycle. And people told us, like, there is almost no chance that you're going to see this. There is no enough resolution because you're just kind of painting the X chromosome. And we were like, okay, let's do proestrus and diestrus, but also over-optimized animals. And we really did not see enough. It was not enough to see changes across the cycle. But it was really important to see that there was increase in the volume of inactive X chromosome after overoctomy. So you remove the ovaries and you see increase in the inactive X chromosome. This is also associated with the high C on this data. It's also associated with the increase in interchromosomal interactions, something that we also see across the cycle. And then there was also increase in some of the expression of these long non-coding RNAs that are important for X inactivation. So basically, these data are indicating that the inactive X chromosome is actually changing the volume across the cycle. And that these dynamics may be important in the adult female brain. I just got like an R21 NH grant for this, so please stay tuned because I think this might be really like a very exciting data. We also look at these enhanced and promoter interactions, and there are around 2,000 of this. And I now want to say that not like people were like, oh, so you see only changes in X chromosome. It's not really true, right? Only in the compartments, but we do see changes in the autosomal genes as well. And what was also important here to show whenever there is like increase in estrogen, they become more similar to males. That's again really interesting to us. And then there was this enrichment for estrogen response element, right? So before I told you there was no enrichment, but now we do see enrichment of estrogen response elements. Just to give you the idea how this looks like, this is one neuronally important gene. You see very different types of enhanced promoter interaction during the high and low estrogenic females stages, and this is associated with changes in gene expression. However, there was something that was very intriguing to us, right? So you see 2,000 of these differential 3D genome interactions, but only 10% of this actually are of differentially expressed genes are associated with changes in interaction, which is really strange because you would actually expect that they are functionally important, right? But if you take all of these 2,000 differential interaction and you do pathway analysis, you see that they're enriched for the brain disorder relevant genes, in particular serotonin and anxiety. So again, we get serotonin and anxiety as a top term. We don't get this when we compare females to males. So there is this idea that maybe there is something that I like to call the molecular epigenetic priming, which means that maybe hormones are actually inducing a lot of changes in the 3D genome, but then you really need another factor such as stress or something that actually make this functional or change it into changes in gene expression. So what I also want to summarize so far is actually based on our toxic and RNA-seq data, we see that probably the membrane-bound pathway is being involved. Based on the nuclear data, we think actually that high-C data, we see that nuclear pathway has been involved. So we do believe that both pathways actually are participating in what we are seeing. So before I go to the second part of my talk, I just wanted to break so make sure that everyone is following so far. So we see extensive chromatin organizational changes across the cycle that at least partially overlap with gene expression changes, which means that they are important for gene regulation. There are compartmental changes in X chromosome, implying something that I like to say that X chromosome is breeding across the cycle, which can actually have really a potentially important role in the implication for female health, and we really need to understand this more. Many chromatin changes are also not associated with changes in gene expression. And this is something that I like to call the molecular priming, that hormones might be actually preparing the genome, but then you need maybe another factor. And what is really interesting is that these differential interaction are enriched from brain disorder relevant pathways. So I know that a lot of people here think about stress. So if you're thinking about hormones are preparing the genome, maybe you need the stress or something to actually push this toward changes in behavior and maybe even psychopathology. So now I showed you all of this, and then people ask me, okay, so this is all done in the cycle, but how do you know that it's really functionally important? Because you can say, changes in behavior correlate with changes in synaptic plasticity, with correlate with changes in gene expression and changes in chromatin. So we needed to ask this question, is this functionally important, right? And we use two different approaches. The first one is the classic approach, which basically people typically use. They remove the ovaries, and then they give you the estradiol and try to rescue the phenotype, right? So this is what we first did. So we basically took our over-optimized animals, give to some of them estradiol, to some of them we gave vehicle. We used the dose that we showed previously can change behavior, so that was important. And then we performed the entire 3D genome assay that I already showed you. It was really quite remarkable to see that there is this, again, X chromosome changes that actually we were able to replicate. Basically, around 20% of the changes that we are seeing across the cycle, we could replicate with estrogen treatment in over-optimized animals. So of course, this is not 100%, but as I said, like estrocycle is not only estrogen, and then there's also a lot of adaptations that are happening after over-octomy. We also look at the enhancer promoter interaction. We were able to actually reproduce with estrogen treatment around maybe 20% of those. This is, for instance, your interaction across the cycle, and then this is how it looks when you give estradiol, right? There was again a very interesting approach when we overlapped the estrocycle and EGR1 and the estrogen treatment. And then we did the enrichment analysis. We again saw the enrichment for EGR1. And this actually brings me to this functional assay that I wanted to actually tell you about. This is a new study. It was not published, but it's actually available as a preprint. It's under revision right now. So we really wanted to try to do something that was not done before, to basically try to model the estrocycle phase by overexpressing the transcription factor, right? And so the idea here is like, instead of giving estradiol, you go downstream and basically you overexpress EGR1. This is estrogen-dependent gene. And also I like EGR1 because also it seems that it's kind of a mediator between the two pathways, right? So Devin actually did his thesis. He took the AAV, like viruses, injected them into specifically ventral hippocampus. We used the promoter that is H-seen, which means that actually you are seeing overexpression only in neuronal cells, because we literally sorted for neuronal nuclei, just to remind you. We did this in over-optimized females now, because we wanted to actually remove the ovaries and like their impact. And then we were able to show that there is only overexpression in the ventral hippocampus, but not the dorsal hippocampus. And the overexpression was around 10-fold, which is, of course, super physiological, but it's not something crazy. And then Devin did the behavior, gene expression, ataxic chromatin organization, and dendritic spine density, everything that I just showed you before within the estrocycle, with this idea, can we model the high estrogenic phase by overexpressing EGR1? And after 3 weeks, after he overexpressed EGR1, he was actually able to show indeed. I'm just showing one elevated plasmase, but this is the same across all the tests, that you see decrease in anxiety indices by overexpressing EGR1 in females, but no changes in males. And this is not associated with changes with overall activity, so it's actually specific to anxiety indices. Then he took, so the changes that we are seeing is actually are proestros-like. Now he took these over-optimized females, overexpressed EGR1, then did RNA-seq instead, overlapped the genes that are overexpressed after EGR1 induction and that are changes across the cycle. 20% of them were actually overlapping. And just to give you an example, this was the same gene that I showed you before, GRIA3, and then these another, both genes are important for synaptic plasticity. You see how they change chromatin and gene expression across the cycle, and you can actually reproduce this by doing overexpression of EGR1. Then there was this important question, if you overexpress EGR1, can you open the chromatin in specifically, right? And I already told you that like we were thinking whether EGR1 can be the chromatin opener. So when we did the ataxic after EGR1 overexpression and did the motif analysis, importantly, the very first actually transcription factor was EGR1, literally showing that more than 50% of opening are specifically happening in EGR1. This is actually, I would say, a first female-specific immediate early gene as a chromatin opener that people have shown. When you look, like, I want to say that EGR1 can open chromatin both females and males, but in different places. And for instance, this is like one of those genes that I showed you previously. You see opening of chromatin in females, but actually not in males. And then we finally look at the synaptic plasticity. After EGR1 overexpression, you see increase in the dendritic spine both in females and in males, but the effect size is much larger in females. So what I showed you so far, you could either give estradiol or you can actually overexpress EGR1 to change behavior, brain structure, gene expression, and chromatin organization in a way that is sex-specific and pro-estrus-like. And so now everything that I showed you so far was done on neuronal cells, right? I told you we sort for new and positive cells. And now what we are doing is actually we want to do this at a single nucleus level. So we are doing this multi-omic analysis of our 3 female groups on the ventral hippocampus. And what is really important here, like all the data that you've seen are all of this, when you put all this nuclei together, now we are going to have this data on the every single nucleus. And this is also going to include not only neurons, but also glia. So please sorry. Okay, so please stay tuned for this because we already have this data and now there is a lot of data. So I think, like in this case, with a single nucleus approach, we are going to have actually the better resolution. We are going to also include the glia. So kind of, as I said, like the data are being analyzed. We have some really interesting new, ideas because there are now more than 30 different cell subtypes. And there are some genes that are changing only in very specific cluster of excitatory neurons or something. So really, we are going to have much more to show soon. So before I finish, and because I'm here at the Douglas Research Center, I do want to talk a little bit about humans. I just want to break here and just to say, like, I hope that I persuaded you that we should take into account ovarian hormone status and that this can change the epigenome and hopefully, contribute to this sex specificity that we are seeing anxiety and depression. And that this can actually help us design more new treatment that are sex-based or sex-informed. I do also want to say again, hormones are not everything, right? The majority of women and other, people that menstruate will not develop anxiety or depression, but it's really the interaction with other factors. And what is really interesting that we have this priming hypothesis that, changes are there, but you may need another hit for something to happen. So now, oftentimes, people can be, okay, you're interested in anxiety and depression, but how does this matter for humans, right? Because this is all done in mice. So the question is how do we make this work more translational, right? And I want to claim something here. I know it's bold, but I really strongly believe in this, that this is a very rare situation in mice that we have a construct validity of the animal model. The reason for that is very difficult to model in mice early life stress and things that are very kind of specific to humans and the human condition, right? But here, what we are talking about is really physiology, right? Reproductive physiology that is very similar in humans and in mice. And really, etiology can be modeled in mice because we are having similar physiology, and we are also seeing something that I already showed you, that there are changes in anxiety and depression-related behavioral indices that are very similar in mice and humans, right? I already told you, whenever you have these big ovarian hormone shifts in humans, you're seeing increase in depression, anxiety indices, and you are seeing this also in mice. I already showed you that there are changes in the brain structure across the cycle in the ventral hippocampus of mice, but there is in humans, there is something that is anterior hippocampus. Is actually, in humans, instead of ventral dorsal axis, you have this anterior posterior axis. And there are more than 70 studies in humans that actually show that there are changes in the brain structure, like a gray matter and white matter across the menstrual cycle. There are imaging studies that were done, and the study of Julia Sacker at actually Max Planck in Leipzig showed that basically these changes are actually correlated very well with the ovarian hormone changes. So it's actually, there is a brain structural change that is very similar. So now we wanted to do something in the postmortem human brain, and I was thinking there's almost no chance that I'm going to now ask brain bank for the luteal and follicular phase in humans, but we were thinking maybe we can actually look at the menopausal transition, because I want to remind you that we saw this really jump in risk during the menopausal transition. 2 to 5 times there is increase in risk for depression, right? And this is an age group that I would say between 45 and 55 that we were actually our targeted group. We wanted to do the cellular characterization to look at the chromatin changes and how they correlate with the mouse data that we got. However, there was one problem. It turned out when I started asking the brain banks that there is no information about the reproductive status in the brain banks, right? So we actually had to go backwards and then first find the proxies for the reproductive state, because we are talking about people that are between 45 and 55, and they could be anywhere, because the chronological age is really not a good proxy for the reproductive status within this age group, right? If you're in 20s or later, like in 60s, for sure. So what we did, actually, I teamed up with Stefano Marenko, who is the director of the NMH Brain Bank. And so Stefano actually sent us the, tissue from the 42 individuals. This was like a frozen blood, plasma, hypothalamus, pituitary gland. We look at the 39 different biomarkers, including the 14 steroid hormones in both blood and the hypothalamus. We look at the FSH. This is something that actually goes up after menopause because it's, the system is trying to actually make the ovaries work. It's actually from the pituitary gland. The AMH is the anti-Müllerian hormone. In humans, is actually used as the ovarian reserve marker for fertility reasons. And then we look at some hypothalamic pituitary gene expression, only several candidate genes important for the reproductive structure. And then this was our study design. We have 21 individuals between 45 and 55. We call them perimenopausal. Then we have 10 individuals that are premenopausal, right? And then 11 that are older than 55 years old, supposedly postmenopausal, right? And so 18 of them were MDD cases. And the idea was we wanted to see how we can find the markers that are going to be premenopausal-like and postmenopausal-like, and then basically classify these groups so that we can study their biology and how things change across the transition to menopause. So I don't want to show you all the data. I'm just going to focus on the 7 strongest markers. You see how nicely the blood estradiol, blood progesterone, and blood AMH actually are decreasing across the cycle. This is perimenopausal, and postmenopausal. We also see increase in blood FSH, so that was all good. When you look at the hypothalamic estradiol, actually, there is a pretty nice correlation between blood and hypothalamic estradiol. This was important to us because some people do not have access to blood, right? So we wanted to find something that could be a proxy in other tissues. And then when you look at the FSH in the pituitary gland, that's also really interesting. In pituitary is such a good resource if you can find it. Actually, you see very nice induction in both pituitary gene expression and protein across the menopausal transition. But if you see, like, there's always overlap in all of these markers. So none of these markers really seems to be completely perfect. So we were thinking maybe we can find a composite measure that can help us actually classify individuals into pre and postmenopausal-like. So we found these 7 markers, right, that they are highly correlated. And then we did a principal component analysis when we found that we can find one component score that actually is a very good representative of the menopausal status. So for instance, like based on this, you can do the cutoff, and this is your perimenopausal group. You can say these are premenopausal-like, and then these are postmenopausal-like. So this now allows us that actually we have now the proxy, and then now we can actually do the actual work. I also received the anterior hippocampus from all these same individuals. So, and we are planning to do a lot of these, studies on the multiome and like 3D genome stuff. But we were also interested. I promise I'm finishing now. We are also interested to see, are there really similarities between humans and mice? So we decided to do something fun, right? So we did actually the multiome analysis in both mice and humans. So this is like your high estrogenic female mouse that I already show you. There are around 30 different clusters. In different colors, you see different neuronal population. This is excitatory neurons. These are inhibitory neurons. And then we took one high estrogenic female. I also want to say when we ask for tissue, we did not ask for tissue only from women, but we asked for the tissue from women and other people with ovaries because we had the non-binary people in our cohort. So why would you remove the non-binary person just because they identify as something that is not women? So I just want to tell you to pay attention when you are requesting the tissue because it can be important for gender inclusivity. So when we look at this high estrogenic human, we were actually able to see a very similar cell types, which is not surprising, but was really nice to see that we can do the multiome with the human tissue. And then we did something fun. We put together human and mouse data and ask them, can they cluster together, right? So basically, and it was really interesting. So in orange, you see human, in green, you see mouse, and you can see that there are clusters of cells that actually are mixed human and mouse. So you can basically tell that there is some overlap between gene expression patterns in humans and mouse. So this basically shows us that in mouse and humans, you have conserved major cell types with some common and of course some divergent features, but also it's very promising to tell us that we should expect to see changes in humans that we are seeing in mice, right? Because there are a lot of similarities also across different phenotypes in humans and mouse. So what did we learn today? Ovarian hormone fluctuation is a major psychiatric risk factor, and the estrous cycle matters for neuroscience. There are extensive chromatin organizational changes across the estrous cycle, functional and priming, right? I mentioned this, with very intriguing changes in the X chromosome. So please stay tuned for this. I feel like there is something really cool about this X chromosome, inactive X chromosome that we are going to discover. we were able to go from our genomic data using physiological mouse model to in vivo mouse manipulation study with EGR, for instance, to validate chromatin regulators and potential drug targets, right? Because this is really cool to know that you can use genomics to find something that actually you can then functionally validate in vivo in mice. Similarities of the neurobehavioral effects of the reproductive cycle in mice and humans are remarkable, but we still need to do the molecular characterization of the postmortem human brain to prove relevant changes in the human brain. And inclusive language generates inclusive data. As I said, we wouldn't have the information on non-binary individual if we did not use the inclusive language. So thank you very much for your attention. I would just like to acknowledge people from my lab, particularly Devin Rocks, who is now at Weill Cornell doing his postdoc, Maria Tikerhova, another postdoc in the lab, Leila Ivana Jaric, who actually started this. A lot of also undergraduate students, our bioinformatics colleagues from Einstein, John Greeley from Texas A&M, Masako Suzuki. 3D genome was done, the bioinformatics part, in collaboration with Jordan Rowley and Mamta Shukla, at Nebraska Medical Center. The NMH, Stefano Marenko and Peter Schmidt for all their help with the menopausal transition project, and also our, collaborators from Fordham, Eduardo Gallo and Henning Cham. And of course, NIH and Brain and Behavior Research Foundation for their financial support. And thank you so much. I would be happy to take any questions.
Question? Thank you very much. That was an excellent talk. And I also want to stand corrected because I know I misspoke your name at the beginning. So sorry, Dr. Kundakovic. I said it Kundakova several times. So this is fantastic. And I'm so glad that somebody's finally looking at this in more detail, because you're absolutely right. We've been looking at everything in males and just assuming that it, can be generalized to females, and that's absolutely not the case. I was particularly interested in one small detail that you showed, which was when you added estrogen to your system. So when your mice were in low estrogen phase and you added estrogen, you were able to recover this behavior right away. But in overactivized females, you added estrogen, but it wasn't an immediate effect. Mm-hmm. Why do you think that is?
Yeah. So this is what we did high C also, for instance, in behavior in overoptimized animals. If you really wait 3 weeks like that is kind of a typical in a way. This is at least what we did. I don't think we will ever do this again. It's because you should deplete your endogenous estradiol. This is what people would say, and then you wait 3 weeks. There's so many adaptations that are happening, right? That you cannot expect that your mouse after overoctomy in 3 weeks, because the body is going to try and the brain is going to try to adapt to all of this. So when we look at the for me, overoptimized animals look like a totally separate. I don't want to say strain, but totally different thing. So I think it's really about the adaptations. Mm-hmm. So you need to kind of restart the system. So it takes some time, but you can do and redo everything. It just takes more time. Mm-hmm. Well, I think the cyclical mice are just so primed to respond to estrogen, right?
Thank you very much. This was an amazing talk.
Thank you very much.
Congratulations. My question will be maybe very naive. You were saying that some of the changes could be not unraveled, but enhanced by stress. Mm-hmm. And would you consider that, for instance, microglia activation in conditions, in disease conditions might be this triggering mechanism or somehow participate in the changes that you see at the level of the chromatin?
Yeah, I think this is an excellent question. I do want to say, as I mentioned previously, that we did everything neuronal nuclei, right? So we don't have, but now with this multiome data, we really are going to have a lot of this information also from glia cells. So it will be really interesting to see what's happening. But also another idea is that we need really to do some kind of studies of interacting, stress and, different phases of the cycle. I think that's going to be really important, because there were studies actually that showed, depending on the cycle phase, if you have acute stress or the dendritic spine density changes are going to be a different direction even. We have studied with early life stress, which I guess I did not show here because there was so much else, but I know that you guys are very interested in early life stress that show us actually, if mice actually underwent early life stress for certain phenotypes, you're losing the effect of the cycle. And for me, this is so interesting because I don't know if this is at the level of that ovaries are not really functioning properly because the animal was stressed and like it never actually starts cycling properly, or there was something in the brain that changed that does not respond to estrogen similarly. But it's like we showed for addiction-related phenotypes, some change in expression that actually once you have the stressor at some point of life, that the cycle has a different kind of effect. So I think these are super important, right?
Oh, no, we have to use the Zoom.
Can you hear me?
Oh, you can't on Zoom. Oh, my, you would want that. So the first question is really pedestrian. I'm really taken with the EGR1, as you can probably
Yeah, for sure.
Yeah. Is there any evidence that the ESR alpha and EGR1 complex?
I don't know. I'm not aware, but as you know, EGR1 is another name for that, NGFIA, right? It's your
Exactly.
subscription factor, right? And that's for me also what is interesting because I feel it's also response to stress. And I think it's really a perfect adapter in a way, right?
Yep.
That like, you know, it responds to, there were also studies that actually showed that EGR1 changes with the circadian rhythm, which is amazing too, right? So it almost seems like it's this master regulator that can actually adapt to differences in the internal, external environment. So I don't know about that, but it's an amazing question. We should really do more about now characterizing this.
Well, you know, the reason I ask is I'm trying to gauge whether estrogen may be working as a pioneer factor in terms of regulation across the landscape. But you've prefaced the next question beautifully. EGR1 also has dynamic effects on neurogenesis in the ventral and anterior hippocampus.
Yes.
And given what you're looking at in terms of both the hippocampus and anxiety, I'm wondering have you factored neurogenesis into any of this?
Yeah,
because that changes over the cycle too. does
Yes,
it not?
so Devin did it, I have to say. He did it, like, you know, he was just leaving the lab and it was done and it wasn't clear. It was, the effect was not there, right? So we tried to do that and like, you know, we didn't find the effect, right? So we did it.
Yeah,
But it doesn't mean like, you know, that, you know, under different conditions, it could not, but under the condition that we did is overexpression for 3 weeks, we did not see for these experimental conditions, let's put it this way. But it's an excellent question. Because there were genes that actually were enriched for neurogenesis and stuff. When you look at EGR1, it's all very logical for sure.
Yeah, and the chromatin, I mean, the exon activation is insane. So
Okay.
we're talking later, we'll get.
Yeah, I am so
amazing.
excited. So we got this grant and like, so the idea is that you need to have this genetic model that can allow you to distinguish between active and inactive X chromosomes. So there are mice that actually have, one of the problems with the X activation is that it's random. Sometimes maternal, sometimes paternal. So it's really difficult to account for this. But there are mice that actually have mutation in X-cyst, which actually makes X-cyst unable to inactivate. So in mothers, it's always active. And then in fathers, you know, like you have activation. So we can control for that. And then you use mice that are genetically very different. They're like kind of wild mice as fathers. So you can bioinformatically do everything that I just showed you across the cycle and stuff, but then you have this information. Was this coming from active or inactive X chromosome? So I'm really so excited about this. We also did some studies of X0 mice. This is also, I don't know if people know about this because it's super also interesting to just know that in mice, only 5 to 7% of genes escape inactivation, in humans 15 to 25%. So there is so much more in terms of escapism in humans that can also tell us that this mechanism actually could be something very important, evolutionary, and also in terms of its dynamism, you know, like in interaction with hormones. So I'm super excited. We just got this grant for this.
That's amazing. So just one last quick point then. If you're suggesting that, you know, there's such a high percentage that escape X inactivation, are you then suggesting that percentage and its nature is dynamic over the cycle?
This is what we are trying. It's my impression, and like, but what also is an important thing is like, if you have increased changes in volume in the inactive X chromosome, you may not be changing only expression of X-linked genes, but you could possibly, because it's been shown that, like, if you have inactive X chromosome, there are all these repressors that are bound. So imagine if you change the volume, you could potentially push this repressor towards, let's say, other chromosomes as well. So I don't know. This is all the question that we need to answer, but it's like we hopefully in 2 years or so you will see. But thank you so much. It's all great question. Thank you.
Thank you so much for this amazing talk.
Thank
I
you.
was wondering if you have some results in the other side, in the other phases of the cycle, and if you do, why you choose this two?
So we still wanted to kind of simplify it. So the question is, like, just to make sure everyone knows, is like, did we look at the different phases of the cycle? So we did in terms of behavior. I showed this data that, like, only during these high estrogenic phase, you have less anxiety indices. But we did not really look across the reason why we chose was the most extreme and, like, kind of the most those that we can relate to different phases of the menstrual cycle in humans. But of course, it would be nice to actually look like at the real dynamics, how everything is looking. Thank you.
Hi. Hi, it was a really great talk. Thank you so much. In your behavioral data, you showed that in like the anxiety test that mice in different phases of the cycle show pretty like marked changes in the behavior. I was curious if those changes are sustained at all after stress, or if you looked at that at all. Maybe I missed that, but I'm just curious if like you think those changes would still be present after a stressor, or do they kind of even out?
Yeah, that is the idea. I think this is what we need to do, right? That we should actually try to see it. As I mentioned, like, someone did this acute stressor, but at the level of dendritic spines, and was showing that there is a completely different direction of change if you stress animals during high versus during the low estrogenic phase. I do also think this is very important for humans. There were actually studies, it's more like anecdotal evidence, that if women, let's say, were raped, sexually assaulted, and they go to ER, and then they get this next day pill, right, to prevent pregnancy, that this may decrease the PTSD, for instance, right, the risk. So even from humans, we have this, like, idea that maybe there is, like, this correlation. One of the problems, and I have to say that very clearly here, like, when people study stress, if you study chronic stress, it might actually change their cycle every day, right? So it's kind of a little challenging to understand, what is but it's still possible to study. You just have to be very careful, like, is it important the day of test or the day when you stress the animal? Like, just because we did like CPP, like this cocaine place preference studies, where it turned out that what really matters is, like, in which phase of the cycle mice were when they were exposed to cocaine for the first time. Right, the first exposure to cocaine, right, mattered. So so I don't know, but it's something that we really have to do, and I would expect differences.
Hello, thank you so much for your talk. It's very interesting. I was very struck by, obviously, your really important result with kind of your high estrogenic, low estrogenic mice, and how they are similar or non-similar to males, even if same age. And I'm just wondering if in this study you took only adult mice, or if you had played around with ages, and how early on in their kind of introduction to having an estrous cycle you would see this difference. And, at like P28, when they're just starting to be on this cycle, do the females, do they have this kind of big separation between them, or do they have to kind of have more of a mature estrous cycle?
Yeah, so it's again another important question. We have studied only young adult females, which are typically between 2 and 3 months old. I do think it would be very important to see when they are establishing, the cycle during the puberty. It's like there might be then some interaction also with stress, and something that actually can affect how the cycle starts being regulated, so I think this is important. We haven't done anything later on. Mice typically do not, let's say, have menopause, but there are models where you can literally inject mice with something to slowly deplete the ovarian reserve. So there is a way to study this in mice, which at some point we should. And I think we should also study postpartum, because I also expect every time when you have these huge ovarian shifts that you should actually see huge changes in, both behavior and chromatin and gene expression. Do we want to respond to this from Munich?
There's no more questions. One last one. Otherwise, we'll thank Dr. Kondakovich for coming today. Thank you very much.
Okay, thank you.